Okay, welcome everybody to this discussion. This discussion is structured around the two-part exhibit entitled Sensorial Skin and Woven by Nature, offered to the Belgian public by the artist Annemarie Maes, who is with us today, and curated by Camilla Colombo, who is also present, and she will talk this evening. Today, this is the first discussion organized by Laser Talks Brussels. Laser Talks was founded in 2008 and now has 47 sites around the world. Leonardo, the International Society for the Arts, Sciences and Technology Laser Talks are a program of international gathering that bring artists, scientists, humanists and technologists together for informal presentation, performances, and conversation with the wider public. The mission of the lasers is to encourage contribution to the cultural environment of a region by fostering interdisciplinary dialogue and opportunity for community building. I would like to introduce to you our moderator of this discussion, and she will be with us for the next Laser Talks discussion, Dr. Edith Doove. Edith Doove, she is a curator, she is a writer and researcher, and she is specifically interested in notion of emergence and contingency, cross and transdisciplinary collaboration. She is a postdoctoral advisor with the Trans Technology Research at Plymouth University, where she attained her PhD in November 2017. And she is a regular contributor to Leonardo Reviews since 2010. Edith Doove founded Bureau Doove in 1914, and she continued to develop a unique, bespoke way of collaborating, working with, for, and alongside artist and researcher. Edith, your turn, please.
Okay, thank you, Alexandra. Very excited to work on this first Laser Talk Brussels, and especially because it's with an artist that I've known for quite a while, Annemarie. You've been active as an artist since 1998, around the time that we got to know each other, I think, well, a bit later maybe. And your work has since been shown in various international exhibitions. As Luc Still in his preface to your book Alchemia Nova of 2016 explains, your work was initially more social, anthropologically orientated, with projects like the People Database from 1998 to 2004, and Politics of Change from 2008 to 2010 that brought you to India. There's then a shift towards ecology and sustainability, situating your work even more between art and science, with projects like the Open Green since 2008 and the projects around beehives, the Transparent Beehive from 2011 to 2013, the Sound Beehive from 2014 to 2015, and the Guerrilla Beehive from 2015 onwards. More recently, you are focusing on yet another collaboration with the non-human, to which we will return later when we discuss your current exhibition, Sensorial Skins and Woven by Nature at PILAR and iMAL in Brussels. What is typical for your way of working is that almost all your work is conducted in these long-running projects. Amin Medosh quotes in his text for Alchemia Nova how feminist science studies observe that the rise of male patriarchal science was based on a particular construction of nature as dead, lifeless matter. In a Cartesian concept of science, nature had to be purified from all myths and symbolisms to become an object of science. This is now, however, being reversed, luckily, amongst others due to the influence of female thinkers and scientists such as Isabel Stengers. Medosh notes that as part of this reversal, you re-inject narrative in the mixture, and possibly a narrative that can also be understood as the call for new stories, the urge for new stories by Donna Haraway, referring to the stories of Ursula Le Guin. Can you comment on the choice for working in long-running projects and their relation to the narrative, or to narrative as a way of working?
Good evening, everybody. Yes, of course, I can comment on this question about narratives. Let's say that, first of all, maybe it's easier if I share my screen and I show some images that makes it a bit more lively for the people. Okay, let's see. Share, okay. Yes. So as Edith stated already, my work is oriented along long-term projects, and one of the first ones was the people database. I'm not going in detail because I want to focus during this talk more on the art science and the bio art. But in the beginning, I was more or less working from an anthropological point of view, and still working, my interests always went to working with communities and minorities. So the people database is a very long-term project that covers many different stages. But it originated in South Africa, where I collected a collection of passport photographs. And I started to work with these photographs as avatars. So I invited a lot of people, first by mail art, later online. I built specific databases, different apps for it to choose an avatar of your choice, and to tell via this avatar, to talk about things that are not talked about normally. Well, for yourself, I mean. So it's about creating one big conscious dream. This took different forms of presentation. Here, for example, in Tour and Taxi, this was in the year 2001, just the right moment that the Twin Towers in New York were bombed. We were building up this exhibition. There I collaborated with a lot of artists on this people database, but also the public had a very important role. And public was always very present, or the actions of the public was always very present in my artworks. Another long-term project was Politics of Change. Here you can see two sides of it. It was about Indian women, but at the other side about European women, commenting on each other and in how they work with technology, with eco-technology, to empower their world. And this empowerment of small communities was and is still a red thread through my art. May it be like women or bacteria or bees, honeybees or whatever, it's always about empowerment. And network, communities and collaborations is another very important point. So this was, let's say, I did many other artworks too, but I will focus now more on the today, and the today for me starts around 2008. In 2008, I started working more focused on ecology. So there was always technology, but now also ecology came into the game. When we started with European project called a laboratory on the open fields that investigated possibilities of building artworks outside but artworks with technology, and not be in the safe space of the white cube or the black box. So outside gives a lot of possibilities, but also dangers. And at that time, I started to work with honeybees and honeybee colonies, but also with urban gardens. And these urban gardens, I called the project Connected Open Greens. The connected, I come back to it later when I talk about the honeybees, but the open greens were for me open blank spaces in the city where no spaces actually that I wanted to turn into green spaces. And therefore I started a collaboration with the honeybees. I started to put beehives on a lot of different places. And one of the investigations was like, can we create green corridors in the city? Urban corridors that is good for wildlife or, well, small wildlife, let's say insects and so on. And can this insect in their turn contribute to these green spaces by being pollinators and as such increasing the biodiversity. So this is one of the long-term projects, and with this narrative of communities working together, enhance each other, empower each other, and so on. This, for example, is, well, sorry that I'm switching so fast, but this is a picture of my rooftop garden in Brussels, which is also my laboratory and my studio. And on this spot, from this spot, everything departed. And on this spot, I also collaborated with a lot of different communities, be it my neighbors, people that just know about the project and asked me like, can we do a little sub-project? Can we do an experiment and this and that? Because there is place enough. It's a rooftop of 2,000 square meters. And now nearly 1,000 square meters is changed into a green source of biodiversity. This picture is from the beginning in 2008. And I can show you a picture from, this is 2 years ago. So it changed into a little urban forest garden with a diversity of mainly fruit trees and herbs and wildflowers, everything that is important for honeybees and for their existence. Yeah, Edith?
Yeah, thank you. Now, Meadows saw you as a model figure for the ecological artist, mainly due to the notion of what he calls a praxis. So it's the idea of a constant life activity, which realizes itself through artistic research, the creation of ecological networks, and different forms and types of cooperation. Now, what does this practice mean on a daily basis? I mean, you showed us your rooftop garden already and gave a hint of that. In other words, how do you work, and specifically, how do you find the balance between art and science?
Find the balance between art and science. I mean, for me, art and science are one in my daily life, but science is a big word. I don't pretend that I'm really a scientist as yeah, with a scientific education. For me, is I'm an empiric person. I learn everything by doing, and I'm constantly, from the morning till the evening, I'm working in my lab, which is my garden, but also my my studio is built on the rooftop in a wooden cabin. And when it's too cold in winter, I just work in my living room, let's say. So everything is mixed. For me, there is no, it's not a 9 to 5 shift and then change and so on. So I have to admit that I also have this luxury of being able to work like that with this rooftop garden and so on. But yeah, for example, when I'm talking about art and about my rooftop garden, once there came the curator in 2013, and he was asking like to recreate my rooftop garden as an artwork in an exhibition. I told him this is impossible because like it's a rooftop garden of 1,000 square meters. How do you want that I do this in the exhibition? And he insisted anyways. And I said, okay, I try. And then I recreated anyways in the middle of winter. The exhibition was running from November till end of February, like the worst moment to have plants and everything. But I managed to recreate at least a part of my garden in this inside room as a kind of biosphere too, because I didn't want to make like an outside garden. I wanted it to be like an artwork on sich. So in a space closed without windows, but where everything was monitored, like a biosphere, and where the visitors could walk in. And I collaborated with the local people, like for bringing me vegetables, for planting, helping to plant, for the maintenance with schools, I collaborated and so on. So it's all based on these collaborations and help. Yeah, well, everything comes out of the real life and then it's turned into art in one moment or another. Here is a small detail of this garden, of the invisible garden, which is called invisible because it was so as a closed, seen as a closed system, not seen from the street or whatnot. But this is a poetic moment. It were small videos that I recorded in my own garden, specific moments, and they were placed there between the plants, and they created a moment of rest and peace. But the main thing I'm busy with is my bee colonies that are also situated in my rooftop garden, but that are also for me co-creators of my artworks. And one of the first works that you see here, it's called the Transparent Beehive. It's part of, again, a long-term running project called Bee Agency, where I want to give back autonomy to honeybees. Everybody knows that honeybees are decreasing, that it's going very bad, and that is at the long run that it will be a problem also for the humanity, for food things and so on. So I decided to do a research on the position of the honeybees and what is known about it with the public. So first of all, I started to build observation beehives. There are two phases in my bee agency project, like the observation, which is learning from the bees, and then later on, trying to find solutions, problem solving, I mean, so proposing solutions. So the Transparent Beehive is part of the observation part. And I built this beehive inspired by Francis Hubert, which is a Swiss entomologist from 1800s. He did a lot of interesting research on honeybees. For example, he discovered that the queen is a queen and not a king, which was until then thought of being a king. And he built this observation beehive in the form of a book. But I took this idea because I thought it was like such a nice idea to be able to stroll through the leaves of a book and read the development of a bee colony, that I transformed it like on the idea of the sliding things in a library, the sliding cabinets in a library. And together with my then colleagues of the artist-run organization OCNO, I built this beehive and we enhanced it with a lot of non-intrusive technology, simple webcams or infrared cameras, infrared lights, so that we could film the bees at any moment. And then we put some contact microphones in the wooden frames and also in the wax to build, so that due to the vibrations, we could record specific sounds of the colony. And as you see, there are two big speakers beneath the sculpture, the living sculpture, and the bees are as such making their own soundtrack with the movie that they make in live. This installation was for 1 year, so a whole bee season, possible to visit in a little laboratory upstairs of our OCNO spaces. And a lot of people there saw for the first time in their life a bee colony from very close. They've never seen a bee colony and how it functions. So people were really amazed by this, how everything works and so on. And actually here the bees, they go outside via the glass, via the plexiglass tube. And in the beginning it was a bit difficult for them, but once that one bee found the way out, all the other bees followed, and there was no problem at all to work like that. So this was a very interesting experiment. And out of this experiment, we collected some, we collected some data, audio data, visual data, and so on, that we later transformed in visual application to put 2 different days of the bee season, or even from 2 different years or from different colonies, the one next to the other. So this, for example, is 1 day and this is another day. And normally it's a movie, of course, it works, but here I don't have the time to put in the data for the moment. But it's based upon audio, on hive temperature, and on the motion tracking of the activity of the bees at the entrance of the beehive. And these 4 wheels that we have here, they, with the audio brightness, they go from blue to white. With the hive temperature, they make the wheels turning, smaller, slower or faster. And the motion tracking, so the activity of the honeybees at the entrance of the beehive, makes that the amount of particles that we see, that they go into a bigger flow or a smaller flow. So actually with this visualization, I can easily, in one blink of the eye, see how 2 days or 2 colonies compare to each other.
Yeah.
Then another project that I did in the garden, and that is a very everyday like way of doing, is looking at your flowers. And then I was wondering, like, where do my bees forage on? Where do they get the food, and do they still have enough food? Is it good for the bees in the city? Do they have enough food in the city? So I wanted to find an answer on these questions. And I collected some pollen that the bees brought back in the beehive. That's easy. So you have a collection of pollen. And then I had some befriended scientists at Brussels University with whom I could work on the scanning electron microscope, a very powerful microscope, where you see the results here in the pictures. And I thought like, okay, I will be able to find online some databases of pollen, and then I can see like, okay, this pollen belong to this flower, and then I know where my bees are going. But it was not so simple as that, because pollen, it's a live substance, and they can differ from one moment to another moment. They can implode completely, or they can be very beautiful. Actually, I was super lucky that I was able to make these pictures without being a scientist and without preparing my samples, because nothing is prepared. It's just pure naked as I collected them. And here you see the apertures, here. But then I was like, okay, I was completely fascinated by this, the invisible. When you go into this kind of world, it's like you feel like Alice in Wonderland. It's a new world that is opening up completely. So starting from this pollen, which later I collected on the flowers itself in my garden, so I knew which pollen were which flowers. And for example, the ones that you see here in the picture is Solidago canadiensis, this one, and this one is Calendula officinalis, both very good bee flowers. But I was then fascinated by the shapes and the forms of the pollen. And I started to use that in my artistic research to like give a solution to the problem of the disappearing honeybees. And for me, this solution was like, okay, I'm going to create new beehives, but in the form of a prototype, but I'm not a designer, so it's not intended to be like a prototype that I want to produce later, but as a sculpture, actually, as a living sculpture. I wanted to create this prototype that is lived by a colony of bees, and that is focused on their needs and not on the needs of the beekeeper that wants the honey from the bees. Yeah. So the bees only like as pollinators and give them the best possible means to live and to develop as a colony. Okay. This was my goal.
Yeah. In that sense, in 2018, your work was presented in the book Eco Visionaries, that presents contemporary positions in art and architecture, seeking answers to current environmental problems that transcend mainstream notions of sustainability. And you referred already in your talk about the bees that you were looking for solutions for problems, or that you made this prototype for the beehive, thinking from the bee rather than from the beekeeper. But do you feel as an artist that you have some sort of task or responsibility to seek answers or find solutions to environmental problems?
Well, see it as a task that's too heavy, maybe. But anyways, as an artist, I am very conscious of the problems that arise, and I think that it's in one way the task then, yes, as an artist, to raise awareness and to work at least with ecological materials, to raise awareness on the problems, about the problems, and to talk to the people in an artistic way and point out what can possibly change, or it's more in this way. And actually, like when I was thinking about building this new beehive or creating this new beehive, well, first I started like reading a lot of scientific papers about the needs of honeybees and what they like the best, the content volume of a beehive, how they want their orientation, the height of the hive in a tree or on a wall, or all this kind of stuff. And then I let go my imagination and I started working like inspired by these forms of nature, like from the pollen, for example. And then also I took a serious interest in working with new materials, because for me this new beehive was like, okay, it's a refuge. You more have to see it like the refuge for birds, but then a refuge for swarming honeybee colonies in the city. And this refuge should be mobile. From there, my name Guerrilla Beehive, because it's mobile. That you can stick it to the wall of a building, you can stick it to the branch of a tree, or on all kind of different interesting places, interesting for the honeybees. And then the swarming honeybees can go and take this place at their home. And when they decide, or if they decide to leave the place, this home should be able to decompose completely, like a rotten apple that falls from a tree and that decomposes. So yeah. I wanted to work with natural materials that can decompose. And here you see that I started in my greenhouse, in the rooftop garden, in my open air lab, to work on the form. But I started also to raise these materials. I read about it online, about these fermentation processes, where bacteria spin cellulose threads to protect the medium of fermentation. And this, by spinning the cellulose threads, they make some kind of skin, of vegetal skin, vegetal leather. I started to work with these processes in depth, reading a lot about it, experimenting with pigments in the mediums. It's a medium of green tea or black tea. Actually, it's like very known in common as a kombucha medium, but I do it a little bit different, because I make the medium for raising the skins and not for making the drink. But here again, it's colonies of bacteria that collaborate in a symbiotic way with yeast cells to make, at the one hand, the fermentation process of the drink, but at the other hand, to make the skin. And here at the right, you see a very thick skin that is colored. It's grown in a medium colored with hibiscus flowers. And you see that it's at least 1.5 centimeter, I think, 15 millimeters. But when it dries out, you have only a few millimeters left. It's 95% water in these skins. But the result is then a real vegetal leather that is really strong when it's so thick as that. And I started to raise them in my lab. Here you see a lot of different containers that where I experiment with different pigments and with different compositions of the medium, because you can steer a little bit like how fast this cellulose will be spun by the bacteria. The warmer it is, the better. And then here, I put a lot of these skin, well, a lot. One, actually on this picture, I put a wet skin in the top of the beehive to see if some bacteria that like this medium, because it's a very acid environment. It's like 4 or 3 to 4 pH. So it's very acid. if I could attract some bacteria, because I wanted to go further, not only this skin that can be the outside of the beehive, but on this skin, I wanted to grow a second skin grown by other bacteria that can sense the pollution of the environment. Because working with a scanning electron microscope, I dissected also some bee parts, and in the fur of the bees, I discovered a lot of little pollution particles. So the bees transport all these particles, and once that they come into the beehive, they transfer these particles to the other bees and so on, which is of course very bad. And I wanted to check the status of the environment of the foraging fields of the bees, like with a natural pollution sensor. When the honeybees come back to the beehive, and when they land on the outside of the beehive, and you have such a kind of biofilm that is made by bacteria, these bacteria, they can react to the pollution that is in the fur of the bees. So the bees will activate the colonies of bacteria with the pollution in their fur. And these colonies of bacteria, they will react with a change in color or in texture of this biofilm. And this was my hypothesis for my next research, where I started to go on the search of the ideal bacteria to work with.
I have to interrupt you there because I'm sure we could talk for hours, but I'm very conscious of the time. And in a way you answered already a bit another question that I had wanted to pose you, but we'll skip it. But on that sort of followed from the article that was published last year in Leonardo with Dennis Dollens on the dialectics of nature and the notion of the dialectic approach or collaborative debate and the work between you and Dollens and others, and in fact the others, or the making kin is with bacteria, bees, and this process of making new materials in a way. But maybe you can slowly but surely also talk about the exhibitions, because Sensorial Skins and Woven by Nature show this focus on membranes that you were just showing, and surfaces, on organic materials, and on tactility, on processes, and on transformation, because you said already that things are not necessarily staying as they are. They can disappear, they can change over time. It's art that stays very much alive and we see in the meantime some images of the bee agency in Hamburg in 2019. You call them a journey into working with natural elements and co-creation with living organisms. And in Staying with the Trouble, Donna Haraway famously talks about the importance of that making kin, symbiosis, or making with, as opposed to autopiosis or self-making. And I think you're commenting actually continuously on this journey and the results of this co-creation. But can you indicate a main difference between the two exhibitions for you? Why have you split them up in two?
Why we split them up in two, I leave it maybe to Camilla Colombo, the curator, to talk about that. Because this was like after a discussion between us or talk between us, like her idea to split it up like that. But I can quickly comment on why and how the making kin and the symbiosis of Donna Haraway and so on, and then come to the exhibition. For example, quickly here we see the living sculpture, a beehive with a real bee population that is positioned in the harbor of Hamburg. It's a commission for, of art in public space. The hive is there now for 3 years and the bees are still very alive. But at the same time, it's a commission and it's an artwork, but it's a living artwork, and I continue doing the research on it. And like here, for example, you see the artwork from Closer. It's inspired on a pollen, and it's milled out by a KUKA robot in the Fab Lab. And I milled out this very specific design on the outside, just for making biofilms of bacteria, of making it easier for biofilms of bacteria to grow on this outside, for then being able to measure the pollution. So this was inspired also on lichen that grow on woods, or mosses that grow on trees. And here algae, biofilms of algae that we see. And then by doing some artistic residencies in, at the University of Amsterdam and also at the Erasmus Hogeschool in Brussels, I was talking with scientists, and they told me, like, you should work for making this, developing these biofilms for your pollution sensor. You should work with cyanobacteria, because cyanobacteria are bacteria that are very strong because they do photosynthesis, and as such they provide in their own food. So you don't have to give them nutrients. They only need sunlight and water. And on top, they take in CO2 and they give us pure oxygen in place. And on this thing is the second part of the exhibition is built, the exhibition at iMAL that will open next Thursday is built completely on this research of working with cyanobacteria that work as plants actually. Here we see like a little essay already of what will be shown, not in this format, but these are chains of cyanobacteria, which are unicellulars, but they present themselves also always in the form of chains. So this is customized blown glass cells with living colonies of cyanobacteria. So actually, the artwork that you see there is purifying your air, just like a plant does. It's the same principle. So this is for the part 2 of the exhibition, and part 1, the one that is still going on for 1 week at PILAR. We have some examples here. I will scroll very quickly through it. It's more focused, so it's focused completely on sensorial skins. And for me as an artist, it gave me a possibility really to work on form, shape, tactility, the senses. You smell, you can feel. It's all about organic materials and about long transformation processes, about the right timing and so on. We see there in the background like the sensorial skins. Also on the table you have real skins, thicker, thinner, skins that are colored in the medium, that are colored afterwards. We see here a wax, the golden beehive. It's a wax sculpture. And these are biofabrics that are made with agar-agar and water, but what gives the specificity is the additives that in this liquid process that I add in this liquid process. You have to see it as tools. For me, it's as the tools comparable to the tools of a painter. So the left one is made with sand, the middle one is made with herbs, and the right one is made with pigments. But also on the second floor of the exhibition, I have many more of these biofabrics that I present in all kinds of hanging, lying, hanging from the ceiling, in light boxes, whatever you name it. And then here, that's my last slide. Actually, that's a new work also that I made for the exhibition, with blacksmiths together, a structure to present the skins in a three-dimensional way. And the one here to the right, it's another experiment that I made in my open air lab. This is 3 meters on 1.5 meters. And it's some kind of serendipity that came out in the form, because it's a skin that I was growing for 6 weeks outside, in the rooftop garden, in a pond that I made especially for that. And it's like some kind of archaeology of the garden. You don't see it on this picture, but it's full of little animals, worms, bed flies, bees, all kinds of stuff, leaves, and so on. And it gives, like, for me, what is an overview of my daily life.
Oh, beautiful. Well, thank you so far, Annemarie. If we find some more time, we can maybe also talk a bit more about the notion of abstraction, the play with form and color. But I would first want to give the word to Camilla. Camilla, you are the co-founder and artistic director of OME, and an organization that develops new practices of scientific mediation, interdisciplinary artistic creation and innovation through collaborative practices. And you worked together with Annemarie Maes on these 2 exhibitions.
Yeah.
So can you tell us a bit more about your work as a curator with, amongst others, OME, and then about your collaboration with Annemarie for these exhibitions?
Okay, I will quickly share my screen too, and I will go very quickly through a few information about OME to give a bit of a context. OME is an organization that I co-founded with a group of friends that are all engineers with a very strong scientific background and all very passionate about the arts, while I don't have any scientific background. But I always had a strong curiosity about sciences. OME is structured in 3 poles, a studio, a lab, and what we call OME Academia. OME Studio works as a sort of production company. We support artists in new production through when they are interested, especially in collaborate with scientists or to investigate particular scientific topics and areas. And we also, through the studio, organize events, and that's where I develop my practice as a curator. I will quickly go through a couple of images. What you see here is a recent experiment of a new production that we are developing with Guillaume Schweicher, who is a postdoc researcher in chemical engineering. What you see now is actually a process of crystallization of biocomponents that change from crystal to liquid, complete liquid when it's all black, and then recrystallize in liquid crystals. We work on this type of materials for audiovisual performances, for example. Sometimes we develop content with a more pedagogical interest. Here, for example, is Sign, that is a sort of interactive lecture performance about the physics of electronic music. That brings in the point of view of the scientist and the point of view of the musician in doing music synthesis and composition. Then there are the exhibitions that can be like the one that I am working on with Annemarie, or that can be like this that you see now in the picture, which is called .img. It was an exhibition that took place last summer here in Brussels, quite literally arts and sciences in the most straightforward sense, in the sense that it was an exhibition that structured as an installation on its own and presented 13 couples of images issued from different laboratories of research of the ULB, who is the Université Libre de Bruxelles here in Brussels. All microscopic photo of microscopy, excuse me, and images issued from the different collections of the Museum of Ixelles, which is a museum in Brussels that focuses mostly on modern Belgian art. The exhibition was an exercise inviting the public to explore and discover these couples of images, trying to move forward the very automatic impulse of finding out what is it that they are looking at, to actually see themselves seeing. As a lot of other people before me said, it was an exercise about perception, visual perception, based mostly on a book by medicine Nobel Prize Eric Kandel about reductionism in neurosciences and in visual art. And then, of course, there were influences by John Burgers and Walter Benjamin. So we bring in many different things. We have another exhibition coming up in June about art and mathematics in Beaux-Arts, which is, again, which the pedagogical aspects and the mediation are very predominant, very important. And then lab, very quickly, it's basically our internal R&D little capacity. We develop it through the lab. What you see here is just a very early stage prototype of an installation that is based on automation and regulation mathematics, and that actually explores the movement of the waves, and that somehow brings in a questioning of how we as humans actually can't really replicate nature if not through technology. Academia, which is the last pole of OM, that is the branch through which we develop interdisciplinary programs for universities and with the universities. For example, Recherche en Perspective, quite literally research in perspective, is a project between the ULB and the Visual Art Academy of La Cambre, who we initiated and we keep and that we manage for the two universities. This project brings in researchers in sciences, applied and pure sciences, with students in visual and graphic communication to work on the concept of science mediation and diffusion. The outcomes can be statics or can be somehow, in some cases, has been quite creative. We had some video animations and like that. We also support and we investigate, we collaborate. For example, in this case, the image that you see here is a photograph of an installation that we co-produced with duo of artists called Bots Conspiracy here in Brussels and the Neuroscience Research Institute, that tries to exploit brainwaves of a member of the public to visualize the changes in the brainwaves when the member of the public is invited to relax and to actually go towards meditation. Last but not least, we are starting timidly to work towards developing the capacity of doing research, transdisciplinary research with the universities, and that's what we are. That's very quickly I think that to go back, I think that gives a bit of a context. To go back about Annemarie exhibitions.
Maybe Annemarie can share her images of the exhibition, or do you have?
I have a few images that I can happily share. Not as many as Annemarie, but they are here. Are they? They're not here anymore. I'm sorry. Annemarie, you have to share your screen, please. Do you mind? The exhibitions, yeah, the exhibitions are two, because we started reflecting, we started thinking about the concept of alchemy, which is our current theme in Annemarie's work. And it was very instinctive for me to suggest the idea of working on volcanic elements, namely the earth and water, which are very distinct in Annemarie's work, in the selection of work that we made for these two exhibitions. The first one, you see a photo here, the one that is currently open at PILAR, in a very instinctive way, recalls immediately to the element of the earth, in the sense that it's very material and it's based on a palette of color that is warm, that is textured, that is as we said a lot, as Annemarie very, very clearly put a few minutes ago, there is a focus on the concept of surface and of membrane, and is strongly characterized by the materiality of the works that are presented. These ones that you see now are a recent production, and then there are the ones on the table. The skins are impressively tactile. When seen live, they smell, they really look almost like animal skins. They have a presence that is not possible to ignore them. They are very terrestrial. They're animal. There is a sort of, I had a thought about the concept of the uncanny valley, of the fact that there is such a strong resemblance with animal skin, with the type of skin that we know, that we touch on our body on a daily basis, and yet we don't recognize it's there, because of course it's not an animal skin, it's a bacterial skin, but there is a sort of sense of confusion in some moments when confronted, especially with certain of these skins. And there is, of course, the element of time, of change over time, of the evolution of these materials and these surfaces that are surfaces very much like the skin. Indeed, they are surfaces of contact. There is a clear intention not to see the membrane as a separation element, a dividing element, but very much as a communication element, something that a material that allows informations to pass through.
Okay.
Yeah.
Sorry that I interrupt. I just want to know, Alexandra, how much more time do we have? I don't hear you, but anyway, I get the idea that we can talk a bit more. So if we, indeed,
sorry,
there's no.
Sorry, I was I unmute myself. So I'm sorry. Just I would like to say that we have to 10 minutes more. Just
Okay.
I think.
Okay.
Sorry.
I'll quickly pass to the next, to woven by nature. There, We don't have photos to show you yet because the exhibition is not open yet, but you saw a few photos earlier about l'origine du monde. Cyanobacteria, why it's the origin? Because it's the origin of life, because it's water, because cyanobacteria requires water, because water is this sort of primordial broth where life came from. And there is a strong shift in the aesthetics, in the visual impact of the exhibition. It's going to be much darker, it's going to be much colder. We pass from a white box filled with earthly colors and warm texture to glass greens, blues, and a taste that is more, excuse me the term, scientific, a bit more cold, even though there is really very little of coldness. There is life pulsating in this glass cell. So there is a shift towards the element of the water that is about the ambience and the general narrative of these two chapters, earth and water.
Yeah, unfortunately, I won't be able to see them as Brussels. It's more than 10 kilometers away from where I am actually at the moment, and I can't travel that far. But in a way, there's again, I had to think of the fact that recently Laurie Anderson, who's giving the Norton lectures, has given an interview saying that in the beginning, you say in the beginning there was water. She says in the beginning there was stories, which sort of seems to make a good roundup of what we've been hearing today. I don't know if there are any questions on Alexandra from the audience.
We don't have questions from the audience, so just like
if there are no questions, I would like to follow you on that, Edith, because I was writing today about the next exhibition, about this movement by nature, and this exact same concept came to my mind. What is extremely important for me about Annemarie's work is that it's a work that allows for storytelling and for narrative, in the sense that it's so multi-layered. It can be appreciated, it can be discovered in so many ways, and each of them has a story, whether it's a story about how she created it, whether it's a story of her rooftop garden that we just heard, whether it's a story of her personal connection with this type of materials, with the otherness that she collaborates with, the otherness in the sense of the non-human, whether it's animal or vegetal or in between like bacteria. There's a lot of stories, there is a lot of narration there, and I find it extremely fascinating. It really opens a lot the way that her work can be presented to the public. It's very fascinating.
Exactly, and I think, and maybe we can give the last word to Annemarie, but I think exactly the notion of the things that change, because I was lucky to see the origin of the world in Paris when it was shown there, but the notion that every time you show your work, Annemarie, it's changing, it might even, as we said earlier, completely disappear, is also something that connects to the notion of storytelling that also continuously changes. I think this storytelling is not static. Maybe you can comment on that.
Yeah, of course. This is for me part of the long-term process that every time that I present it, it's in a change, and I like it to be that way. And even if one day it would completely disappear, for me it's not a problem. Like it's like life itself. For example, like the skins that we saw lying on the table in the exhibition at PILAR. Mm-hmm. They were mainly made from 2016 till 2019, let's say. But the skins, they are aging just like a human skin. In the beginning, they are completely fresh and very supple. And then due to environmental parameters, mainly to UV light, but also to the humidity in the air, or being non enough humid. It makes the skin like tanned, and it makes it feel and look older, with wrinkles and all this kind of stuff. And I think this is a very interesting fact, because all this makes that every time you present it, it's changing. There is an update, but yeah, once again, it's an update like life itself. So I think for me, that's one of the main goals that I have with my art, and what interests me in making art.
Yeah.
That it evaluates, it evolves with life.
So your work is in a way also not destined to the museum, that is more conservative in
Well,
ways.
I don't agree with that, because there are so many things that are not destined to the museum, but the museum often kills artworks, I think. Like, look what they did with Joseph Beuys' artworks. Do you really think that Joseph Beuys would have wanted that the fat would have been remodeled every time, again and again? And no, he was like, of course he wanted the same thing, like that. Let it go. Let it be itself. Let the material be itself. Give power to the material also, I mean, and not to the curator, or or not a curator. It's not a curator, maybe, I don't know, but the people that decide that it has to be always the same. I mean, it's a pity, no?
In that sense, there's a connection, I think, between your work and that of Eva Hesse, that is slowly deteriorating as well.
Absolutely. Eva Hesse is one of the artists I like the most, for her sensibility, for her experiments that she did with new materials. It was often with not so natural materials. She experimented a lot with chemical resins and so on, but at that time it was like fabulous that she, as a woman, she was the only person that dared to go out of the usual. I mean, and she created a beautiful minimal art with these new materials. I like very much her work. Yeah.
Okay, I think just we are come to the end of our discussion, because we a little bit extended the time, but I would like really to thanks everybody who took part in this discussion: Edith, Annemarie, Camilla. Thank you very much for Leonardo and Lazar Turks to help us and just to be with us, because I see you are present. And thank you very much for Silent for this technical support. And I think that we will be soon back with another discussion that now we are preparing with Edith. And thank you very much for being with us this evening.
Thank you.
Thank you, Alexandra.
Thank you. Well, have a good evening.
Bye.
Okay. Well.